I'm Professor Maggie Smith from the University of York, and I've always worked on the interactions between bacteriophages and their hosts, which are members of the genus Streptomyces. The Streptomyces are gram-positive soil microorganisms that are really important in biology because they make lots and lots of interesting secondary metabolites, including the all-important antibiotics. The Streptomyces make lots of interesting bioactive molecules, including the antibiotics, but they also make things like anti-cancers and other therapeutic agents. So we are interested in, of course, generally people who work on Streptomyces are interested in why the Streptomyces produce these things, but also in how we can understand when and where they're produced, but also how we can manipulate those pathways. So we have been working on the viruses or the bacteriophages that infect these organisms with a view to understanding more about the organisms to answer the question why and how, but also with a view to generating the tools through which we can manipulate those pathways. In other words, we can move the DNA that encodes those pathways to make the antibiotics into other Streptomyces or other organisms to make them better, to make them different, to make them more effective, and to make more of them. In phage-host interactions, the phage can sometimes reveal a lot about the biological processes that are occurring in the host. And the reason for that is because the phage are parasites that get inside, the DNA gets inside the cell, and then they basically reprogram that cell in order to make more of themselves. And to do that, they have to be intimately connected with all the biological processes inside the cell. And for every phage-host interactions, those connections between the biological processes and the phage replication, they're always different. The phage are literally a window on what's going on inside the host. And of course, I've already said the host is a really interesting organism. It makes antibiotics. And so we can use that phage to find out more about what's going on inside that host, and therefore hopefully manipulate that host to make more antibiotics, if you see what I mean. And in terms of mechanism and why it's important to understand mechanism, we don't want to just sit around and observe stuff. We want to be able to understand why and how things work. We want to, as scientists, we see something, we ask why and how and where and when, and so we do experiments to test those and think, well, maybe it occurs when this happens. We do an experiment to test that, and if it's right, we can ask another question and test something else. If it's wrong, then we leave that and move aside to something else. What most excites me, I think, is that opportunity to ask how, where, why, what, and the opportunity to be able to do experiments to test those things, to answer those questions. I think that's the most exciting thing. So it's basically following your curiosity. And what I find really satisfying is when you make an observation that maybe have passed you by, and then you see something and you think, oh, that's new, and that's what's really exciting. Well, when I found out I'd got the Marjory Stephenson Prize, I was so delighted and so shocked and surprised. I couldn't believe it. I stood up and my knees practically gave way from under me. I was almost in complete biological shock. So I was, delight is the answer. I was absolutely delighted and totally honored, feel completely honored by the society to be standing up there giving it the Marjory Stephenson lecture after all those other brilliant scientists have given the same lecture. Yes, I'd love to thank everybody who's ever worked in my group. So all the people I've had, PhD students, postdocs, visitors, technicians, and undergraduate students as well. I really want to thank all those people who've dedicated time, effort, more, beyond what was necessary to do the research, and they have been equally excited about my research. I would also like to thank my collaborators who've worked with us to answer specific questions, because occasionally I end up asking questions that are outside of my own skill set, and so I turn to collaborators, other academics who have the more appropriate skill set. Funding bodies, of course, they are absolutely essential when you're a PI. You can't do the research without the funding. But I've had some really important mentors during my career, and so I'm going to be thanking those mentors in my talk. The most important of whom is the late great Roger Hendricks, who really helped me bridge that almost a gulf between Streptomyces community and the phage community, and that was really important for me.
I'd like to welcome the Marjory Stephenson Award. It's an award offered to an individual who's made exceptional contributions to the discipline of microbiology. And this year's winner is a truly worthy winner, Maggie Smith. She's an emeritus professor at the University of York. Her work has led to discoveries in the regulation of phage development, the evolution of double-stranded DNA phages, a new system for phage defense, a Streptomyces protein glycosylation pathway, and the mechanism of a novel group of phage integrases. Maggie probably doesn't remember this, but I actually published a paper with Maggie. I won't say how long ago it was, but it was with some bald guy called Paul Hoskinson, who was in short trousers then. Anyway, please join me in extending a very warm welcome to Maggie, whose lecture is Bacteriophage-host interactions in Streptomyces. Thank you, Maggie.
Well, thank you for the introduction, and huge thanks to the Microbiology Society for awarding the Marjory Stephenson Prize to me. The Microbiology Society have always been there for me ever since I was a PhD student, actually, and through postdoc and PI in various institutions, and so I'm very grateful to the Society in all its different forms as it's been through its evolution. I want to thank also my nominators right from the start, who are Drs. Rebecca Hall and Alison McFadyen. And they are members of the ever-expanding ECR group of microbiologists. So, and I'm very, very grateful to them for nominating me. And I also want to thank, at this stage, the institutions that hired me. So I started out at the University of Stirling, and then moved to Nottingham, and then to Aberdeen, and then finally to York. And every single one, I want to give thanks because they saw that I could contribute something, and they had faith in me to give me a salary, basically. So, bacteriophages. I haven't always been in love with bacteriophages. In fact, as an undergraduate student, I found them quite hard. All those anti-terminators and far too many operators. So it was really a decision that led me to work on phage rather than a calling. And that decision was manifested in several different areas and several different reasons. So first of all, there's an enormous evolutionary force going on between bacteriophages and bacteria. And where there is very great evolutionary forces, there is always the opportunity, or the generation, I should say, of biological novelty and diversity. And it was actually discovering some of that biological novelty that drove me. Secondly, bacteriophages can be regarded as a a window on host processes, on basic host processes. And I want you now to think about all the processes that were discovered from Lambda and E. coli interactions. So think chaperones, think the mechanism of sequence recognition by proteins, think mechanisms of DNA recombination. So third, phages can be regarded as a genetic toolbox for the manipulation of their hosts. And so here I want you to think T7 RNA polymerase, think regulatable promoters, think gateway, coming back to phage lambda. And fourthly, it was circumstances and fascination that led me to be working with Streptomyces. But I needed a research niche, and phages were going to provide that niche for me. So when I started as a PI, this was really the question that I posed in a very general sense. Can we find biological novelty in Streptomyces phage interactions? And Streptomyces are gram-positive bacteria. They're really interesting. They grow as a mycelium. The mycelium grows into the soil. They sporulate, and they make a wide diversity of secondary metabolites, including the all-important antibiotics. And the phage system that was available at the time and that what I was going to be working on was called Phyce31. And this is a temperate phage. And it looks like this, and a bit like lambda, so nothing out of the ordinary. But it had been developed by Keith Tater at John Innes Institute already at that time as a cloning vector for Streptomyces. So just to summarize the types of projects that we had going on in the lab, and Del has actually done some of that already. We worked on, so my sort of home, if you like, is protein DNA interactions. And so I started out looking at a very unusual repressor system in Phyce31. We tried to understand about the transcriptional circuitry, which we defined, and a little bit about the activation that goes on during the lytic phase. We did quite a lot of sequencing of phage genomes, which back then wasn't as easy as it is now. And so we sequenced Phyce31 and several relatives and several other Streptomyces phage, and delved down into the genomes to see what we could find that was interesting, again, looking for biological novelty. We came up with many hypotheses, but we couldn't prove them all, unfortunately. But, you know, the information's in the genomes. We worked on a Streptomyces phage defense system called phage growth limitation, or PGL, which is a sort of Baroque methyl-specific restriction system, if you like. Talk to Paul Hoskinson about PGL if you want to know what that means. But PGL turned out to be a forerunner of a much wider group of phage defense systems called BREX. And we've worked on phage receptors, and we've worked on we've done a vast amount of work actually on phage integration and excision. The two projects I want to talk about in this talk are really quite different to each other, but they exemplify different aspects of phage-host interactions. So the phage receptor project is a project, so I should say to start out, all the projects that we worked on were curiosity-driven. We always sat there and said, what if, why, how, and so on. But the phage receptor project is an example of observation, but it also took me right out of my comfort zone. And the integration project, phage integration and excision, is a project that led us somewhere that we really, really didn't expect to go. So with that in mind, let's start the first story, which is the host cell surface recognition by Phyc31. So this is a critical step in phage infection. So if the phage fails to recognize its host, that it's going to be capable of replicating inside or persisting in terms of as an integrated prophage, then it fails. It fails completely. You can't repackage, as a phage, you can't repackage its DNA that it's just injected into this non-host, if you like, and go back and try again with a different bacterium. So it's a critical step. So it has to recognize its host. So as I said, Streptomyces are gram-positive. We knew at the time that we started this project that most gram-positive receptors tend to be sugar molecules on cell surface macromolecules. But we didn't know any phage receptors for Streptomyces at the time. And so the hope was that we'd find out something about the Streptomyces cell envelope by trying to find the phage receptor. Given that most receptors are sugar molecules, we expected to find a glycosyltransferase. So we made some mutants, spontaneous mutants, and we did a whole series of complementation using the ordered Cosmid library that was available at the time. And we eventually found 2 genes that would complement most of the mutants in our collection. And the first of these genes was a homologue of I can't see the laser. Anyway, the first of these genes, its closest functional homologue in the database were the yeast PMTs. So these are protein mannitol transferase that glycosylate target proteins on a serine or threonine. So they are basically glycosyltransferases, as we expected, but they glycosylate a protein. And this came as a huge shock to us because at the time, the textbooks were telling us that prokaryotes don't have glycoproteins. So we were confused, but we did a lot of reading, and we found that there were some reports coming out in Mycobacterium tuberculosis from people working on Mycobacterium tuberculosis that they have glycoproteins. And also, there were some reports from Streptomyces lividans, which is a close relative of Streptomyces stelicola, that lividans also has at least one glycoprotein. So we thought, well, maybe this isn't so weird after all. And the second gene that we found that complemented a few of our mutants was in the database, the closest homologue was a yeast dolichol phosphate mannosynthase, which makes the sugar donor, which in yeast makes the sugar donor for the yeast PMTs, or protein mannitol transferases. Okay, so we named these genes PMT and PPM1, which stands for, because Streptomyces bacteria don't have dolichol, instead they have various polyprenols. So we named the second gene PPM1, which is a putative polyprenol phosphate mannosynthase. So what do these proteins do in Streptomyces? So I'm going to describe this pathway. I just want to say right from the start that we collaborated with Dell, Bezra's lab in Birmingham, in order to show some activity of these proteins. So it's not just an extrapolation from the yeast system. So we actually isolated membranes and demonstrated, with his help, the transfer, the activity of PPM1 and the activity of PMT in a cell-free system. So what do? So PPM1 is a membrane-associated protein that transfers the mannose from GDP mannose to a polyprenol phosphate located in the cytoplasmic membrane to make polyprenol phosphate mannose. And this molecule is then flipped so that the mannose is displayed on the periplasmic side of the membrane, and then the PMT, the protein mannose transferase, takes that mannose and transfers it to a serine or a threonine on a target protein. So we had to, at some stage, demonstrate that we could find glycoproteins in the Streptomyces membrane or cell envelope. And we did that, and we discovered a protein, PSTS, that was glycosylated. And we did this with the help of Andel at Imperial. And we demonstrated that PSTS, which is a phosphate-binding protein, is glycosylated with a trihexose. So PSTS is actually a very abundant protein in the periplasm. We thought, well, that's a good candidate for a phage receptor. So we knocked out the PSTS gene, and the PSTS knockouts are still phage sensitive. So PSTS, at least on its own, is not the phage receptor. So then we dug a little deeper, and we asked, well, how does GDP mannose get synthesized? And so we looked at this pathway from mannose 6-phosphate right through to GDP mannose, and this was mediated by the MAN-B and MAN-C genes. And we knocked these out. In the case of MAN-C, we had to knock them down because MAN-C is actually essential for Streptomyces coeliacolor. So we knocked down MAN-C and we knocked out MAN-B. And these mutants were phage resistant. So now we have a whole pathway that goes from mannose 6-phosphate right through to the generation of glycoproteins that's required for the phage receptor. So, right, okay. Well, it seems then that our model is fairly straightforward. O-glycosylated proteins are receptors for Phyc31 infection. So was this the end of the story? No, not quite. Because we observed that the mutants, all the mutants that we'd made, PMT, PPM1, MAN-B, MAN-C, they all grow pretty slowly compared to the parent strain. And we asked why that. We wanted to know why. So we first of all looked at morphology, because Streptomyces is a morphologically interesting organism, and O-glycosylated proteins are in the cell envelope. But we couldn't find any change in morphology. And those of you who work with Streptomyces, you'll know that one day it looks different from another day, and so this took a few weeks. No change in morphology, so we tried a different approach. And that different approach was to challenge with antibiotics to find a weak spot in the cell physiology. So we challenged with all sorts of different antibiotics that act intracellularly, extracellularly, and we found that the PMT mutant is hypersensitive to ampicillin, well, basically beta-lactams, vancomycin, and oddly, rifampicin, which is an antibiotic that acts intracellularly at RNA polymerase. But the other antibiotics act, obviously, on cell wall biogenesis. So with the PPM1 mutant and the MAN-B and MAN-C mutants, we get even greater susceptibility to the same set of antibiotics. But in addition, we have susceptibilities to daptomycin and niacin, which are membrane-acting antibiotics. So a bit of a more severe phenotype, if you like, for the PPM1 mutants. So we wanted to see if we could find the physiological basis for the antibiotic sensitivity. So we did an RNA-seq experiment on the PPM1 minus strain compared to its parent. And to summarize a huge amount of data, we basically saw massive downregulation of membrane and periplasmic gene expression, implying a major change in cell envelope functions. And we saw a big upregulation. In fact, we saw one of the most highly expressed genes compared to the parent was an ECF sigma factor. And we wondered whether that could explain the rifampicin sensitivity in this strain. So this change in gene expression in periplasmic genes really knits in or ties in well with the sensitivities that we're seeing to the beta-lactams, vancomycin, and membrane-acting antibiotics. And I should stress that we didn't see much change, apart from rifampicin, we didn't see much change in intracellular acting antibiotics. So just a word about vancomycin then. So these are distaffusion assays. I'm sorry, I should have explained that. So Streptomyces silicola has got a vancomycin resistance gene cluster that's been well characterized by folk at JIC. And this cluster is similar to the VAN-A, VAN-B gene clusters that we see in enterococci. So the simplest explanation then is that the gene cluster isn't being expressed in the mutants PPM1 or PMT, okay? And that's why they're sensitive. So we did qRT-PCR experiments to look at the RNA in response to subinhibitory vancomycin, and we found that the amount of upregulation in the mutants was exactly the same as the parents, so there's no change in the RNA expression. And presumably there's no change in the expression of the proteins either, but something is blocking the delivery of the resistance. So you get the RNA, but no resistance. We get the proteins, we presume, but no resistance. So presumably something is blocking the delivery. So in order to dig a little deeper into this, we isolated suppressor mutants of the sensitivity. So these are mutants that have gone back to being resistant, like the parent strain. So, and we did that by picking colonies that grew in these zones of clearing. And yes, we saw straight revertants of PPM1 and PMT. We saw mutants that were massively upregulated in the vancomycin gene cluster because they had mutations in the regulators, VAN-S or VAN-R. And, but we also saw mutants in a gene cluster which is coding for a putative extracellular carbohydrate. And this putative extracellular carbohydrate appears to be interfering with resistance. We don't know how, but what we do think is that, and we haven't investigated this, but it does appear to represent a chink in the armor of the vancomycin gene cluster, and maybe that information could be used to regain vancomycin sensitivity in other bacteria that have got the VAN-A, VAN-B gene cluster. So what about the glycoproteins themselves? What sort of proteins are glycosylated in Streptomyces silicola? So we collaborated, so this was work done by a PhD student, Tessa Keenan, in York, and we collaborated with Andel, again at Imperial, and the York Technology Facility to identify glycoproteins from Streptomyces silicola. And we did that by looking for glycopeptides prepared from Streptomyces silicola membrane fractions, glycopeptides that were modified with either a hexose, a dihexose, or a trihexose on a serine And we found over 90 glycopeptides, which resolved themselves down to 38 glycoproteins, and 33 of these are periplasmic secreted or membrane proteins. 8 of these are glycoproteins, so they're possibly involved in peptidoglycan biosynthesis. We knocked out 2 of these genes, 2 of the genes encoding the genes for peptidoglycan biosynthesis, and they gave an antibiotic-sensitive phenotype, but they're still phage-sensitive. Okay, so those two that we knocked out, plus the PSDS, they're not. None of them on their own are the phage receptor. So to wrap up this little story, we know that 5C31 uses this pathway right from mannose-6-phosphate right through to glycoprotein synthesis to recognize its host. We assume that it uses a whole multiplicity of glycoproteins, possibly even just the sugar or the short sugar chain. We'd like to know how the glycosylation affects the activities of Streptomyces cell envelope proteins, because clearly there is an effect on activity. This kind of information is really hard to come by, and in other systems, and I'm thinking here now about N-glycosylation in Campylobacter or in the yeast PMT system, O-glycosylation is important for protein thermostability. And in Campylobacter, in the N-glycosylation system, it's also important for protein-protein assembly. We also know that in Mycobacterium tuberculosis, glycoproteins are important for adhesion to the host cells. So this isn't an easy question to answer. So that's a challenge out there for you young ECRs out there. So I explained to you that the PPM1 MAN-B MAN-C mutants, which are upstream of the PMT step in this pathway, their phenotypes are much more severe than the PMT minus mutants, the mutants that only do the glycosylation. So, and they are used, those genes, those proteins rather, encoded by the genes PPM1, MAN-B, MAN-C, are all used to generate this molecule, the polyprenyl phosphate mannose. And so this is the sugar donor, and because of the severity of the phenotype of the PPM1, etc., we think it might be used in another role in Streptomyces coeliaca, and we don't know what that role is. We know from Del Bezra's lab that PPM1 is used to synthesize lipoglycans, lipoarabinoglycans in Mycobacterium tuberculosis. But Streptomyces doesn't have lipoglycans, or at least they haven't been found. I'm not even sure they've been looked for. So more broadly, we know that looking in the databases, homologs of the protein aminosyl transferase are present in most, if not all, actinobacteria. So this is a very common pathway. The glycoproteins are all there. They're just there for the discovery. We know that analogous mutants, PMT or PPM1, in Mycobacterium or Corynebacterium are either extremely poorly or lethal. So, and certainly in pathogens, that might be maybe a good target for knocking down the pathogens. But more importantly, this is a loss of fitness that we see in receptor mutants, and it's seen time and time again in phage-host interactions. So it's not just in pathogens that you see this loss of fitness. And it makes perfect sense for the phage, because so for the bacterial host, the easiest way for it to become resistant to a challenge by phage is to make a receptor mutant. And, but of course, when the challenge by the phage becomes less, in making that receptor mutant, the host becomes less fit, then they will get overcome by phage-sensitive bacteria again, and the phage can then have another go, if you like. So it also bodes well for phage therapy, as we've seen people talk about earlier on this afternoon. Okay, so complete change of gear now. And I'm going to talk about, in this second section of my talk, I'm going to talk about phage integrase and how it's been used in genome engineering. So in this part of the talk, and this brings me back to much more to my comfort zone, which is protein-DNA interactions. So we're looking at this reaction, which is going on in order to establish lysogeny, in order to exit from lysogeny in the induction phase to go back into the lytic cycle. And just to explain to you some of the nomenclature and the processes involved. So when phage injects its DNA into its host cell, in the case of Phyce31, it has cohesive ends. So these come together, and host cell ligase ligates them to form a circle. So we have the circular phage genome, and on that phage genome, there's a short sequence called the attachment site. And the phage attachment site is called ATP. And it site-specifically integrates into the equivalent attachment site on the bacterial host. And this is called the ATB site. And the protein that mediates the recombination at these two sites is called an integrase. And after the integrase has done the recombination, and it does it at a specific cis-al phosphate in the DNA, in both partners, in both ATB and ATP, it forms reciprocal products called ATL and ATR, which are hybrid sites, half B and half P. And between them, you have the integrated phage genome. So integrase does this integration reaction, but integrase also does excision. And, but integrase usually needs an accessory protein, which is phage encoded. I should also say the integrase is phage encoded as well. So you need integrase plus the accessory protein to do the excision. And then the phage genome can come out. It again becomes a circular genome that can then enter back into the lytic cycle. So this is back in 1991, so Stu Kustos and Nagaraja Rao, Eli Lilly, published the sequence of Phyce31 integrase. But more importantly, from an applications point of view, they published a paper showing that you could make integration vectors that are derived from the integrase and ATP site from Phyce31. So these vectors essentially are this, except that it doesn't contain all the other phage genomes, if you like. So it's only, the only phage components are the integrase gene and an ATP site. And the rest of the components on the vector are things like a marker for selection in Streptomyces, a replicon for persistence in E. coli, but a replicon which is inactive in Streptomyces, and so it can only persist by site-specific recombination in Streptomyces, an origin is transfer for conjugation. They showed that these vectors are stable, there's no excision because there's no accessory protein, and they go into the ATB site, and they are present in low copies. So I should say you'd expect it to be a single copy, but in practice you sometimes get 2 copies in. So these integration vectors were a stroke of genius by Eli Lilly because they really simplified and made it easier for Streptomyces researchers to clone DNA into Streptomyces. You could make your constructs in E. coli, you just whack them into Streptomyces by conjugation, you select, and you've got a nice, stable integration at a consistent site, and it's in low copy number. So we wondered how we could improve on these vectors. So here's a diagram of the Streptomyces coelicolor chromosome, and this is the location of the Phyce31 ATB site in the chromosome. So the answer to that question, can we improve, is quite simple. We just make another vector that's compatible. So we'd by this time sequenced another phage, Phyce31,
a relative of Phyc31 called Phybt1, which incidentally to the Streptomyces people out there in the audience stands for big turbid one, which describes the type of plaques we were getting. So the integrase looked like the Phyc31 integrase, but it looked sufficiently different that it might have a different at-B site, and we found this to be the case. So we made a suite of integration vectors from Phybt1 with different markers, a flavor that would suit people, and like the Phyc31 vectors that were made by Eli Lilly, we sent these all over the world, and everybody seemed very happy with them. And so we were pleased with that. So why give up on a good thing? So we eventually generated some more vectors made from some different phage. They're not all phage that will actually infect Coelicola. So SV1, for example, is a phage that infects a different strain of Streptomyces called Venezuelae, but it still has an at-B site in Streptomyces coelicola, so we can still make an integration vector from it. So we made all these vectors, and in order to do a proof of principle experiment, we collaborated with Matt Gregory from isomerase, and they had great expertise in a biosynthetic gene cluster for erythromycin from a strain called Saccharopolyspora erythraea, which is a relative of the Streptomyces genus, but different enough such that when you put this cluster in Streptomyces coelicola, it doesn't normally express. So we identified 4 genes that we could isolate in our different vectors, and these 4 genes together should make this polyketide scaffold in Streptomyces coelicola. So the 4 vectors that we made have all got a Streptomyces coelicola promoter upstream of them. And I should say these genes, these polyketide synthases, are huge genes. They're about 10 kilobases in length. This 4th one, a hydroxylase, is quite short and a reasonable size. So we put these 4 genes into 4 different vectors, and we got the erythronolyte B, which is the polyketide scaffold out. So again, we were happy with that. So to explain the next part of the story, I need to take you back to the sequence of Phyc31 integrase as it was published by Stu Kustos and Nagaraja Rao back in 1981. So when we looked at the sequence, it really didn't look like a phage integrase that was similar to the whole vast group of phage integrases that had been published that were similar to lambda integrase. It looked very different. In fact, it looked like a group of site-specific recombinases called the serine recombinases that contained resolvase and invertase. So these are short proteins, about 180 amino acids, and they all contain a catalytic domain and a small DNA binding domain. And that catalytic domain was present in Phyc31 integrase. So we actually knocked out the serine, the active site serine, and we lost activity of the Phyc31 integrase. So the difference, though, is that Phyc31 integrase had got a huge C-terminal domain compared to the resolvases and invertases. And this domain, which we later characterized, contains the DNA binding domains, but it's also required for control of directionality, i.e. that integration versus excision. So we went on to characterize the activity of Phyc31 integrase in vitro. So we purified the enzyme and used it in vitro recombination experiments to find out exactly what properties it had and to compare it with lambda integrase and with the small serine recombinases. So first of all, we found that the recombination sites are both short for ATB and ATP with 5C31 integrase. Now, this contrasts with lambda, where the ATP site is over 100 base pairs, and it uses sites that accessory proteins bind. So ATP and ATB, 45 to 50 base pairs, and the integrase binds as a dimer with the crossover site in the middle. So it's a bit like a repressor-operator interaction, except that it cuts the DNA and does the recombination. And this is the bit that really intrigued us. So integrase on its own, just purified integrase and the substrates, it only does ATB and ATP recombination. Now, integrase binds with equal affinity to all of these sites, B, P, L, and R. But it only does P and B. If you give it L and R on its own, it doesn't do any recombination. If you give it P and P, at P and at P, it doesn't do recombination. If you give it B and L, it doesn't do recombination. It's purely site selective. It only recombines B with P. But it does that recombination on substrates where the B and P are on different molecules, but B and P can also be on the same molecule in inverted repeat, in which case it gives you an inversion, or in direct repeat, that's what these arrows mean. In direct repeat, it will give you a deletion. So the terms integration and excision become a little bit confused when you put B and P on the same DNA molecule. And the point is that it's topologically insensitive. It doesn't require a specific topology in the DNA or where the sites are to allow recombination to go ahead. And I should say that with B and P recombination, you can, in vitro, you get up to 90% products. So the third thing that we characterized was that the reaction going from L and R together to regenerate P and B needs a recombination directionality factor. This is the accessory protein that's phage encoded. And that binds to integrase to activate L and R recombination and inhibit B and P recombination. So it switches integrase from a BP enzyme to an LR enzyme. And the differences to lambda integrase are essentially that it uses a serine rather than a tyrosine as the nucleophile that attacks the DNA. The recombination sites are both short. It doesn't require any accessory proteins required for P and B, and there's no specific topology required. So the conditions for 5C31 integrase to give you efficient integration are really very, very simple. And we published this. And then somebody read our paper. And that somebody was Michelle Callos at Stanford University. And what she showed was that you could use, she was already working on site-specific recombinases, looking for one that would work in human cells. So it was like a perfect situation for her because she took 5C31 integrase and did an experiment to show that you could get site-specific recombination in human cells. And essentially, this was the experiment she did. So she put what's now called a docking site into episomal DNA and then brought in an integrating plasmid with an ATB site and something expressing integrase, and she got integration. And that was what she first published in this paper. It seems that 5C31 integration system is highly portable because then there came reports of people using 5C31 integrase in all sorts of systems. And the one I just want to point out is the Drosophila community, who really embraced the 5C31 integration system. So expressing transgenes in Drosophila can be a bit of hit or miss because those randomly inserted transgenes can find themselves in a place that's in the genome that's affected by position effects, in which case you you don't get any expression of the transgene that you just put in. So what they did was they created transposons with a docking site, one of the attachment sites, located in the transposons, and then isolated transposons in sites that were neutral for position effects. So then you could use integration vectors to bring your gene of interest into a known neutral site in the Drosophila chromosome. And they liked it so much, they renamed the system FlyC31. So we had a collaboration with William Brown at Nottingham during my time at Nottingham. And so what we did was, initially, it was a thorough scrutiny of FlyC31 integrase activity in eukaryotic cells. We wanted to demonstrate the conditions in which the genome engineering aspect of FlyC31 was giving a bona fide site-specific recombination reaction, and those conditions which gave an interrupted recombination which led to small deletions. So we did that, and then William went on to generate a novel method for assembly of very large DNA tracks in an artificial chromosome, and he demonstrated that you could assemble a 400 kilobase pair insert, for example, in an artificial chromosome based on the human Y chromosome. But the experiment that we did that moved everything on a little bit, by this time, there were quite a lot of serine integrases or large serine recombinases out there in the literature, and people were using different ones in their systems that I just described. And we wanted to do a simple experiment to determine which serine integrase is the most efficient for use in human cells. And it piqued me a little bit to find that the answer was an enzyme called BXB1 integrase, which has its origin in Mycobacteriophage. And we demonstrated that BXB1 integrase was the most efficient of 15 candidate serine recombinases for the integration of DNA in the human genome. And FlyC31, incidentally, came a pretty close second. But nevertheless, that information was out there. It was there for the community to embrace. And then moving the whole story on, bringing it up to date, I should say, I came across this paper while I was preparing this talk. And I have to say, it blew me away a little bit. So one of the problems with having to put a docking site in is that that docking site tends to go in at a random position, because you usually put it in by some sort of transfection technique, and it goes in randomly. What this technique does is it gives you the opportunity of pushing your docking site into the place that you want that docking site to be, so you can program where that docking site goes. And it works like this. So these researchers at MIT, what they did was they made a fusion protein between Cas9, a reverse transcriptase, and BXB1 integrase. And they transfected the gene encoding this fusion protein along with two guide RNAs and a mini circle that contains your gene of interest and one of the attachment sites for BXB1. Now, how it works is this. The nicking guide recognizes the desired site in the human genome, and then the @B guide then primes, and the reverse transcriptase primes the addition of the @B site into precisely the desired site in the chromosome, so that you now have a docking site present. And then the BXB1 integrase can then integrate your gene of interest by using a BP @B recombination to give you the gene of interest, which is now flanked only by the at-L and at-R sequences, so short sequences. And you use a mini circle in order to avoid all that vector nonsense that you don't need. And the mini circle, incidentally, is generated in E. coli using 5C31 integrase. So this gives you an integrated gene. They showed that you could get up to a gene up to 36 kilobase pairs integrated by this means. So this isn't the only paper that have done fusions between Cas9 and BXP1 integrase, by the way, or serine integrases. There are others out there, but this one just blew my mind. They also identified over 25,500 other serine integrases from the databases, along with their ATB and ATP sites, with a plan to screen which ones are even more active than BXP1 integrase. So I think that brings me to the end of my story about the applications for genome engineering. So just to wrap up this section, we believe that the large serine recombinases are useful for integrating large DNA fragments into the eukaryotic cells. We did a huge amount of work on the mechanism of the serine integrases. And if you want to know how they work, then please message me on the Microbiology Society app, and we can sit down and we can talk together. So the integration vectors really made a massive difference to research on Streptomyces biology. And I'm talking really about the Eli Lilly vector that first came out here. And so we're very, very grateful to them for having that idea. There are other uses for serine integrases that I haven't been able to talk about, and they've been to devise bacteria that can perfectly switch on and off. And if you string them all together, you can make counters, bacteria that can count, or you can make logic gates, which I haven't, so I haven't had time to talk about them. So finally, I want to acknowledge everybody who's worked in my lab over the years. And this is all the people who've worked, visitors, PhD students, postdocs, and all the undergraduate project students that I've had. And I want to say thank you to them. And I want to say apologize to those whose work I haven't had time to talk about. I'm looking at you, Paul Hoskinson. And yeah, thank you. I want to say thank you to my collaborators, Del Bezra and Del, for the collaborations that I've described today, and also William Brown. I want to say thank you to the University of York Technology Facility as well, who've been great for analyzing helping us to analyze the glycoproteins. I want to say thank you to 3 people at the John Innes Center who've been really important to me, Keith Chater, Mark Butner, and Merv Bibb, for help with Streptomyces biology, Streptomyces strains, phages from Keith Chater, projects and ideas for projects and general support. Finally, I want to thank my mentors. So Ian Hunter, who used to be at University of Glasgow, is now at Strathclyde. It was his lab that I joined at the time when I was transitioning between being a postdoc and getting my first PI job. And he gave me the opportunity to think about what my research niche was going to be. And to my mind, this is the most difficult thing as a young PI. What are you going to spend the rest of your life on? Or even what are you going to spend the next 5 years on? And he really helped me, and he actually provided some funding in the lab so that I could generate some preliminary data that would lead to my first SCRC grant, now BBSRC. So, and then when my first position was at the University of Stirling, and I want to thank Mick North, who was a senior lecturer there at the time, and he guided me through the ins and outs of being a lecturer, which was not trivial, and so he was invaluable at that time. And then after 3 years, I moved to Nottingham, and Bob Lloyd was there. He was head of department, and he was wonderful as a mentor. He read all my grant applications. He always found time to criticize them and to help. He also read some of my papers, and we had great discussions, scientific discussions. He was an enormous help. But most of all, I want to thank the late, great Roger Hendricks. So Roger was a professor at the University of Pittsburgh. He guided me through all of the phage meetings that I went to, and he taught me about the Lambda back catalogue, which was so useful. He introduced me to many of the Lambda biologists in the United States, which was invaluable to me to getting a quick recap on that background of phage biology. He was a collaborator. We worked together on phage evolution, and he was a great, great friend, and he's sorely missed. So on that note, I want to finish, and I want to thank you as an audience. Thank you.